Hello everyone, today we are going to discuss on the topic Biotechnology Principle and Processes Part 1, and this video is presented to you by www.examhe.com. So the topics we are going to discuss in this video is first of all in basic introduction, then principle of biotechnology, steps in gene cloning or recombinant DNA technology, the tools used for gene cloning and nomenclature. Okay. So first of all, we will discuss about the basic introduction. So the term biotechnology is derived from the fusion of biology and technology. So you must be thinking about what are the role of biotechnology in our lives. So these techniques are very useful and we come across to it in our daily lives. For example, biotechnology is used in making bread, making curd, wine, and all these processes require microorganisms. So the bread, curd, and wine, these are produced in very large industrial scale. So these require a microorganism, and the process is called fermentation. The bread, curd, and wine is produced by the fermentation done by the microorganisms. Okay. The importance of biotechnology to human welfare would be the production of monoclonal antibody and development of DNA vaccine, recombinant protein vaccine at large scale. So to understand that, first of all, we have to understand what are the vaccines. So when we fall ill, some bacteria or viruses attack over our body. Okay, and our body produces, like these are the bacteria and viruses. So they attack on our body. Our body, in response to this, produces some immune response. Immune response, they will produce some antibodies. Okay, so the bacteria and viruses will produce proteins which are harmful to our body. Okay, so they attack to us by this protein, and in response to this our body will produce antibodies. Okay, so what happened that uh, because it was a sudden attack, so the our body is not able to produce so much of antibodies that it could survive during this attack. So what happened is we get ill, and all our antibodies are not all our soldiers, or we can say, are not so much well prepared that they could resist the attack by bacteria and viruses. So what happen is, these are the proteins which are artificially made, okay, by scientists, artificially produced, okay, and these proteins are then injected in our body, okay, in very small amount, very low amount. So now our body will consider it as a attack, so our body will produce the antibodies. Okay, and it can survive the attack now. And what happen is, after which these antibodies get stored somewhere in our immune system. Afterwards, when the actual bacteria and viruses attack, okay, afterward when the actual bacteria and viruses attack on our body, these antibodies which are stored somewhere in our body will come and uh, fight with these protein produced by bacteria and viruses, and thus they win. Okay, so likewise this is the use of vaccines. Okay, so when the newborn baby is there, and we used to take him every 2 to 3 months to the hospital for vaccines. They have a full course of vaccines like hepatitis vaccine and other kind of vaccines. So these are those vaccines. So we are actually developing the immunity inside the baby, so that when the actual attack of bacteria and viruses will take place, the baby could easily survive that attack, and could easily be could not get ill. Okay, so this is what the vaccine role is. Now, what is the use of developing DNA vaccines? So these proteins are where these proteins are developed inside the bacteria and virus. These proteins are developed at a very small part of the DNA, which is called gene. Okay, and these gene produces these proteins, and so if we will produce we will use these gene in this recombinant protein vaccine. So the vaccine would be produced, and DNA vaccine would be produced, which can be used further for the immune response. Okay, now next is production of variety of biochemicals ranging from alcohol to antibiotics. So the biochemicals which are produced by many organisms, many fungi, many bacteria are there, which produces a large enormous amount of biochemicals, and these are used in many industrial processes. Okay, now processing of foods. So these biotechnological processes are used in food industry also, and also for the production of enzymes. So this is the whole process which occur. Okay, the next is in vitro fertilization and embryo transfer technique. So this embryo transfer technique and in vitro fertilization permitted the childless couples suffering from one of the other kind of sterility to have their own babies, and these are actually the test tube babies. So what is sterility is not able to produce um, a male or female gamete by male or females respectively. So what they do is, like for example, if we take the ovary produces the egg is then taken out from the ovary and at in this sterile environment. Sterile environment means uh, free from microorganism, free from any dust, and free from everything. It is a very neat environment in which the fusion of egg and sperm take place inside the petri dish. Okay, so this plate is called petri dish. Okay, so what happens is after that it is incubated, and at this time of incubation this fusion would be called zygote. It divides into 4 cells, and after that it divides into 8 cells, and likewise 16 cells. So after which it is inserted inside the female uterus, after which these cells will adapt the internal environment of uterus and will start developing by its own. And when the baby will develop after 9 months, the baby will be healthy and fully developed. Okay, so this is the in vitro fertilization. Now in agriculture, so what occur in agriculture? The production of virus and bacteria free crops, also the genetic engineered crop which would be resistant to the insects, also have developed the improved variety of crops. So what they do is they insert some sort of genes inside the plant DNA. So plant is also living organism, so they also have DNA. Every living organism has their own DNA. Okay, so what they do is they insert some sort of, like this is the plant DNA, so they take out a little part of it and make this half empty, and then insert some sort of gene from some other source, this gene from some other source, which would produce some sort of protein which would be harmful to these bacteria, viruses and insects. So what happen is, when this plant DNA will start producing this protein, which is very harmful for bacteria, virus and insects. So when the bacteria, virus or insect will attack on the crop, they will literally die by themselves. Okay, so this is what happened by the help of recombinant DNA technology. So this is the basic introduction. Now we are going to discuss the principle of biotechnology. Okay, so genetic engineering technique to alter the chemistry of genetic material, either DNA or RNA. So as I have told you earlier that any living organism, they have their DNA and RNA or RNA. So if we alter its configuration, if we alter its chemistry, then that alteration is done by the technique which is called genetic engineering. Okay, so we have seen the advantage of asexual reproduction over the sexual reproduction, in which genetic information is preserved without any variation progenies. These processes also done by genetic engineering, which include recombinant DNA technology. So this is a kind of asexual reproduction type of process, in which, so what happens in asexual reproduction, the genetic information which is present inside the parent cell will be transferred to its progenies. There would be no any kind of alteration, because the progenies are developed from the single parent, not from the fusion of male or female. Okay, so that's why, so this process is also done by genetic engineering. So recombinant DNA molecule produced by joining together two or more DNA segment, usually originating from different organisms. So what they do is, actually, like, this is a host plasmid and this is a site of cleavage. So from this site they cut it, okay, by the help of restriction endonuclease. This we will discuss further. And after that, now they cut this site and will develop a sticky end, and after which the specific genes, which is from other source, from other bacteria or from other virus or plant or any other, so from other source, a specific gene is uh, inserted inside the host plasmid, and hence the recombinant plasmid DNA is developed. So this is the point of attachment. So these are genes which are obtained from the other source, and this is what the host plasmid. So it can also be said that a recombinant DNA or rDNA molecule is a vector. Example like plasmid, phage or viruses, into which a desired DNA fragment has been inserted to enable it cloning in an appropriate host. Okay, so these are the vector like plasmid, phages or viruses, in which a desired gene of interest which we require is inserted into it, and then they are inserted into a host which can produce that gene. For example, if we take, like, this is a plasmid, okay, and if we want to produce some sort of specific protein which is very useful for human health, and this specific protein we get from some bacteria. Okay, so what we do is, we take this gene of a specific protein from the DNA of that bacteria. So this is the specific protein, and this is the plasmid we have as a vector, sorry, plasmid. So this plasmid is a vector. Okay, now what we will do is, we will cut a specific site from it. Okay, we have cut and by some restriction endonucleases. Restriction endonucleases are actually the molecular scissors. They are useful in cutting these DNA. Okay, so now this site will be empty, and now we will insert the gene, specific protein gene, which we have obtained from some DNA of bacteria, and then we have inserted in this site. Now what we'll do, we will insert this whole plasmid inside a host, which would be E. coli or any other. So most common is E. coli, and we will insert inside it. Now this E. coli will uh, produce its progenies, and hence this DNA will be transferred from one generation to another generation with this specific protein gene of some bacteria. Okay, so this is the whole process which occur. Okay, so the vector containing DNA segment to be cloned called DNA insert. Okay, so whatever we have gene of interest is called DNA insert, introduced into a suitable organism, usually bacterium. So the host we have taken is E. coli, that is a bacteria, is organism called host. Okay, and this whole process is called transformation. So what we have discussed here, this whole process is called transformation. Okay, so the transformed host cell then selected and cloned the recombinant DNA present would replicate the gene present in the vector, may or may not express itself. So as we have seen in this diagram, the replication of this gene will take place, but it might be possible that this E. coli would not respond towards that gene, but it can be possible that the E. coli would respond to that gene, and this gene would replicate inside the E. coli and would be transferred to its progeny or daughter cells. Okay, this transformation of suitable host with recombinant DNA and cloning of transformed cell is called DNA cloning and gene cloning. So this whole process is called gene cloning or DNA cloning or recombinant DNA technology. But the bacteria which whose plasmid has been DNA has been altered with some other uh, DNA of some other source, that is called transformed host, and that process is called transformation. Okay, now steps in gene cloning or recombinant DNA technology may be classified into the following 5 steps. So these 5 steps are the main steps which occur in gene cloning or recombinant DNA technology.